Last week, close to 175 stakeholders from across the global fatty liver community convened to hear 43 speakers address a range of NASH and NAFLD issues at the Fourth Global NASH Conference. Join the entire SurfingNASH crew and guests in discussing issues related to improving and using diagnostics in this special conversation from the NASH Congress, today on the Surfing the NASH Tsunami podcast.
Hi, this is Roger Green, host of the Surfing the NASH Tsunami podcast. We are offering one conversation from last week's Fourth Global NASH Congress, focusing on discussions from episodes 21 and 22 covering diagnostics. These issues ranged from discussions of exciting new diagnostic techniques in development to broader questions of when to use these tools and what to expect from them. All 7 people who participated in our coverage had things to say on this topic. Each one makes an appearance in this abbreviated recording. So, sit back, listen, enjoy, learn. And when you're done, join the conversation on our LinkedIn and Facebook discussion groups.
To get us started, Manal Abdelmalek and Louise Campbell discussed 2 sets of presentations, each of which addressed non-invasive tests, but from very different perspectives.
I think what's striking is the robustness and the innovation by which the biomarker development field is moving forward. That is incredibly striking to me. We've been hinged on imaging biomarkers. We've been hinged on blood-based biomarkers. We've really evaluated the strength of even bedside risk assessments such as FIB4 and NAFLD fibrosis score or APRI. And what we're starting to see is a transition to the assessment of point-of-care, potentially biomarkers, whether that be urine assessment, proteases, or even breath tests for volatile organic compounds. And the one promise is if we are able to achieve really sensitive and specific point-of-care biomarkers, as simple as a urine test or a breath test for this disease, that would be key, not only with regards to potentially lowering cost of biomarkers, where you're not performing very elaborate, high-cost, throughput omics, metabolomics, lipidomic, proteomic type platforms. And I do anticipate those costs will come down with time, certainly, but really empower the primary care physician to do an office-based test. And I think what we're seeing is we're getting closer to that. The challenges, of course, that exist, fascinating presentation that I thought that was presented by Gillespie Fernandino, evaluated the AL biomarker, which was a breath test that assessed literally over 1,000 volatile organic compounds in the breath. And they evaluated patients who had cirrhosis and cirrhosis with liver cancer, and even patients who had healthy controls. And granted, it was a very small pilot project, but they were able to discern is there was one volatile organic compound, limonene, that was detectable in the breath of patients with cirrhosis and patients with cirrhosis and liver cancer, although there wasn't much difference between cirrhosis and cirrhosis with liver cancer compared to the controls. And as they drilled down and presented their data, what they ultimately noted is that there's correlation between this limonene and other markers that are known to be predictive of liver function, such as bilirubin or prothrombin time or even albumin, suggesting that not only do we have a point of care, but one that could predict synthetic function in reserve. As I was listening to the presentation, my thought is, wow, this is very exciting, but really how much such markers could be confounded by other disease conditions, whether it be bacterial overgrowth of the gut, or whether they could be even heart failure or dietary composition, high fat, high sugar diets. We know that these volatile organic compounds can change, and they could potentially change over time and not completely be reflective of underlying liver disease. What would happen if somebody took recent antibiotics, for example? So a lot of unanswered questions. I think they're just starting to scratch the surface of the potential for volatile organic compound breath testing, but I think it opens up a Pandora's box also about really assessing how real world these type of assessments can be when you weigh into it dietary changes, medications, exposure to antibiotics. Would they really be reflective of underlying liver disease or other metabolic milieu in general for patients. But I thought it was still very innovative that a breath test could stratify health from cirrhosis, irrespective of the presence or absence of liver cancer, and really correlate more so with a marker of function. But I think there's a lot in store for these type of technologies, and time will tell what we see.
Yeah, so both Suneil and Manal highlighted the biomarkers, and I totally agree. I think it's this part of science that's rocketing here and really moves forward. So I'm very convinced this is going to be one of the major breakthroughs in the near future. And Manal, the story you've been telling, I'm thinking back to a paper I saw at AASLD once where the dogs were actually able to smell, I don't remember, either it was cirrhosis or cancer. And it's so obvious that you're able to quantify those changes now using those technologies. So I strongly agree. And while you're saying there's variability, it can also speak to personalization. So maybe when we understand better what drives which compound, we could then use this to personalize the whole approach and treat persons based on that. So I agree with both of you. It's a fascinating area.
And then the other thought I had was really just a kind of a simple, silly one, right? But we asked people to do fasting glucose. We asked people to do fasting lipids. I could envision a point at the future if it turned out that you could control the variability where we asked people to do fasting breath. It's a funny thought when you think about it, but it's not completely out of the question if that's what you needed to do to control better. Now, Manal, I take your point. Breath is so much more environmental than anything else is, and what they're talking about is so specific. But all this is possible. I guess we have to see where it goes.
It was. If I misspoke, it was from Alstone Medical. So I think it scratches the surface for the potential of point of care biomarkers, and really just opens up a wealth of opportunity by which to look at the robustness of these volatile compounds as they would otherwise perform in the real world. With various confounders introduced, because certainly the last thing we need in a very highly prevalent disease is a point-of-care test that lacks any sensitivity and specificity based on environmental dietary exposures. However, they are able to detect differences between endogenous and exogenous volatile compounds. So maybe with time and additional modeling, we can tease out a more optimal sensitivities and specificities for what really matters, and what a doctor needs to be able to ascertain is beyond just the presence of cirrhosis alone. So a lot of work to be done, but very exciting work.
That was the owl stone. Did you have any reactions to the Glimpse Bio, to the urine-based?
I think the Glimpse Bio is looking at urine proteases, and really I think the same concepts and principles apply with these new technologies. While the platforms are different, the innovation is high, the risk to reward ratio is also very high. And at the same time, I think we need to define with more granularity how these biomarkers perform across the spectrum of disease, whether they in fact can serve as predictive risk stratifiers for outcomes, which is ultimately what we care about. But both, interestingly, are teasing out unique markers that are altered in the context of metabolic disease and metabolic risk.
Yeah, and NASH Tech 2020, where we met, one of the comments I made there that you actually and I talked about later privately is the idea that the winner in diagnostics is going to be low CapEx or no CapEx. The capital expense ultimately is going to be the enemy of primary care treatment. But if these things work out, then you actually not only have no capital expense, but you have low intrusion, right? People aren't going to have problem breathing, and only relatively few will have problem with urine. So you've now got the ultimately patient-friendly delivery of test materials, assuming, as you point out correctly, that they can then be interpreted relatively free of environmental factors. Suneil, let's let Louise go next. It's later in the day in the UK than it is here. So, Louise, what would you like to cover off on today?
I did 2 sessions. One was Lauren Costera, who was the lead session for the day, who's Professor of Hepatology in the University of Paris. And the second one was Andreas Gier. And I apologise now to any Germans, Jörn, who's the Professor of Internal Medicine and Hepatology at Würzburg University in Germany. Now, both of their conversations looked at non-invasive technologies and the state of the art now, and basically into screening in primary care and who to screen. So I'll start first with Lauren's session this morning. So he basically did a very nice summary of the current position of non-invasive technologies, concentrating on a number of points. Can they diagnose NAFLD and NASH? Can they replace biopsy? Can they be used in the real world? And can we enrich for that? So when he was looking at diagnosing NAFLD, he focused on CAP, which is obviously a component of FibroScan, and MRI-PDFF. So ultimately, the advantages for CAT, it's point of care, it's widely available, but it's not as widely available looking at most of the sessions today as people tend to think. Looking at Andreas Gier's presentation, only about 24% of people access FibroScan, and it's primarily where it's located. But it does concomitant liver stiffness, but its disadvantages, obviously the quantifications are suboptimal for the CAT. There's a lot of discussion of where 5%, 33%, 66% steatosis markers are, and they're all quite high. They're all in the 300 range realistically in a US multi-center XL probe study and also in the UK multi-center XL probe study. So they put that against MRI-PDFF, which obviously has great advantages. It's highly sensitive, it's precise on steatosis, and it is the gold standard of non-invasive technologies. Its disadvantage is the time consuming, it's very limited in availability, and it's a high cost, and obviously not a lot of people can get through it. But head-to-head, CAT is well outperformed by MRI-PDFF, and I don't think there's certainly any argument from me on that. But CAT was promising in that it was rapid and standardised steatosis point of care, despite its suboptimal. But there was no consensus on the cutoffs, but above 275 decibels per metre, we get a 90% sensitivity. So that was really quite nice. When he then went on to looking at diagnosing NASH, he reviewed the obvious biomarkers and the scores, but made the conclusion that currently none of these are available, are sensitive or specific enough to really differentiate NASH from simple steatosis. Transient elastography and MRE neither can confidently discriminate NASH from steatosis either, so biopsy remains the reference for that. So we moved on to diagnosing advanced fibrosis. Obviously, we're all keen on the F3, F4, which is what we focus on in our clinical trials. So the serum markers had reasonable AROCs, and most of the validated serum markers for F3, F4 in NAFLD, FIB4 and NASH fibrosis score was simple, freely available and widely available. Had AROCs greater than 0.8 and a high negative predictive value of 90%. ELF was patented. It was slightly higher performance, although there's less external validation and limited availability. So FIB4 non-fibrosis score versus ELF. The ELF was slightly better, but again, lack of widespread application, and it's limited by its cost and availability. They then went on to review elastography, and it's the technique of choice for point-of-care testing. It's available, it's less expensive. It's also one for me that, as I talk a lot, it's about interaction with patients and how they really interact with their condition and their own care. But the high negative predictive value, but suboptimal for F3, F4. But he then looked at redefining Bovino with elastography, and the recent Papathilodoridi in Journal Hepatology paper this year did an optimal cutoff with dual cutoffs for chronic liver disease, and that was a liver stiffness of less than 8 and a liver stiffness above 12.5 kilopascals was actually a better way to use a dual cutoff to define serious disease. So when he summarised it, despite FIB4 non-fibrosis score and ELF, VCTE, so FibroScan and MRE were the most accurate to validate for assessing F3, F4. Use in clinical practice, various different pathways were looked at into how we do first-line care in and around, but also using sequential non-invasive therapies for that. Can non-invasive technologies enrich trials? A very strong prediction for the FAST score, which we know is CAT plus liver stiffness plus AST, work done by Phil Newsome. So that came out higher than IS4 on that, but only marginally. Prognosis, ELF and VTCE were all able to predict prognosis of disease over time periods, and FibroScan was also able to pick up mortality. So in all of the areas that we want to be looking non-invasively, these technologies do work. So his take-home messages were ultrasound remains the first-line tool for detecting NAFLD. Liver biopsy will remain the first time for diagnosing NASH. All of the other are accurate and validated for diagnosing advanced fibrosis. And in clinical practice, the optimal strategy is sequential use of FIB4 NASH fibrosis score to rule out F3, F4, followed by FibroScan realistically. Now, a lot of that was covered again by Andreas Greer, but his presentation was way more focused on how do we use this in primary care and what are the advantages to that and the obstructions. So he looked at NAFLD screening, which generally isn't done despite 20 to 30% of the population's affected. He identified some screening algorithms and the flow through primary care, also diagnostic algorithms and specialised secondary diagnostic platforms. But I think he summarised a lot of what Laurent Castera did. He also focused on what we discuss regularly, that you cannot use abnormal liver function tests to identify patients predominantly at risk, so serum ALT elevated. But what he did go on to, which I thought was an excellent series, was he looked at NAFLD screening in the populations and used two studies, one in Italy, which was more of a diabetic population, so it wasn't general, and another one that was a UK population that was way more of a general population. And actually, it was significant for one reason, that he identified that 30 to 40% of patients is all we pick up through any of our normal techniques with liver disease, so that we are really, really missing a significant number of patients by not screening. He then went on to look at cost effectivity for screening with transient elastography and the number of needed to screen to make it cost effective. And it was as low as 7 in patients with diabetes. It was it was 12.5 in patients with alcohol. In NAFLD, it was 34.5 patients, and obesity, it was 8.5 patients. So the argument for not screening, I think he showed extremely well that we can do it. We don't need huge numbers to be able to screen. And I think if we look at some of the other areas of numbers needed to treat, it would come really highly. Again, he looked at through primary care and made the same conclusion that a lot of presentations I listened to today, that screening and availability to primary care physicians to transient elastography would be extremely helpful. He used the Camden and Islington pathway to go through some of that, but they've designed their own pathway whereby they used a questionnaire with primary care at the beginning. Then they would go to risk scores, FIB4s, and have a fast-track system for FibroScan. And what they said on one of the questions that he was answered, that actually significantly removed a number of referrals by doing it that way and then doing virtual appointments. So that, I thought that was a really good one. They didn't state, however, if they used the FAST score, and over 80% of patients were overestimated if they were bariatric, which I thought was quite interesting.
Suneil Hosmane discusses 3 presentations on diagnostics, and then triggers a controversy around how good NITs must be as compared to medications.
First one was by Steve Williams at SomaLogic. I've met Steve years before, and it's really nice to see his platform evolving just at a super high level. They've found a way to do really high throughput proteomic analysis, not using standard proteomic equipment. Normally you have to do mass spec and kind of use expensive large equipment. He's able to kind of scale that down and use something called aptamers, which is, I'm oversimplifying, but it's a piece of DNA that's been kind of shaped in a particular way, and it binds things, like it bind proteins, and you can start to generate signal on various proteins. And that was sort of one of the big takeaways of the day is, with a very sophisticated system, looking at the whole universe of proteins, or a very large universe of proteins, finding a panel that's around 37 proteins to kind of really delineate steatosis from inflammation, ballooning, fibrosis, NASH, and the OROC, very good, but achieving that 0.8586 range. So from a blood-based biomarker standpoint, given the reference standard, it seems like that may be approaching a limit of possibility. So that's useful and helpful for the field in general when we're thinking about comparing and things like that. And it also tells you that there's a law of diminishing returns as you start to complexify the panel and you increase the cost. There's only so much you can eke out in terms of performance. So it's just an very interesting talk, great data. I'm looking forward to seeing more of that generated through trials. Second talk was on microRNA. So this is Rui Castro. But great talk on microRNA and kind of giving kind of a state of the art in terms of various microRNA biomarkers. And I think one thing that's just interesting is the liver is, as you know, a metabolic factory. It's a protein factory. And as a result of that, it's a biomarker wonderland, so to speak. And sorting through that is challenging. And from a blood-based standpoint, it seems like microRNAs have strong plausibility there. And there's been some great work work. And I think he raised a great point at the end, which is we really need to think about how we standardize these analyses so we can really begin to compare and then move the field forward. And they're little nuanced points, but they're very important things, like how do you normalize a measurement? What's it in reference to? And those type of things, will begin to get ironed out. So a great overarching talk. MicroRNA, the takeaways, microRNAs, particularly 122, 34A, and I think 19 or 21 had significant association with NASH and features of NASH and fibrosis. So more to come there, I believe. And the last talk was from Andrew Billen from Gilead. And I applaud Gilead for, in general, as a company, like no matter whether positive data, negative data, they're publishing it and they're getting it out there and they're moving the field forward. I think we should all congratulate them for that kind of stewardship. And I think the most compelling thing that came to me from that data, I mean, there's many ways you could dissect all that data, but it was a very early slide and it was these Kaplan-Meier curves. And if you haven't seen those curves, they're usually time is on the x-axis, y is like events or survival or something of that nature. And you see kind of the curves with differing levels or ranges. And what I thought was exceptionally interesting there was, we already have data in the field for example, transient elastography, like people with stiffnesses above 38.6 have the worst outcomes versus 12 to 38 versus 8. Well, I'm oversimplifying, or less than 8. And you have something similar with ELF, and within our own program, we're trying to generate something similar as well. And the blueprint for migrating clinical development from biopsy to NITs is right there. That's the strategy. So, for example, if you enroll patients that are above 12, hypothetically, and you bring them below 8, that should be predictive of outcomes, and that's what these graphs show. And I think as we collect more data and we move that forward, we have that discussion with the FDA, I think them being fairly objective people would be at least conversational about that. So I think there are strategies to move forward. I don't know if the right conversations are being had with the people that have the data. So that's an interesting thing. But they went through all kinds of analyses in Stellar, and they were showing how from a lens of a biopsy, efficacy is X, but from the lens of an NIT, maybe efficacy is 2 times X, and that's a provocative way of thinking because the NITs can kind of tease out more of the biology or different elements of the biology than the biopsy can. My last point, which is probably the most controversial, which is why I left it to the last part, is we have to be a bit introspective and say, is it the NITs that aren't good or is it the drugs that are not good? Because if I put out an NIT strategy and a drug doesn't achieve it, does that mean my NIT sucks? Maybe. Maybe that your drug doesn't work, right? Or it didn't work the way that you thought, or you didn't dose it high enough, or you didn't dose it long enough. And I think, if we just be philosophical for a second, let's say it takes hypothetically 7 years to go from stage to stage, yet you want to rapidly bring it back one stage in 6 months. What do you think your chances of success are? I just put it out there to say maybe the NITs are not so bad, but maybe what we're asking of them is too much.
A couple thoughts. So first of all, it's a shame that Manal has left. Louise, I wonder if you know where I'm going, because Manal's been pretty consistent about this issue, that drugs should get approved simply because they stop decline. That, in fact, FDA has made clear that if you maintain and don't regress, particularly in terms of steatosis, that's enough. So that would be in essence, that would align with what you're talking about. I think it's probably fair to say that the historic burden we've asked the drugs to carry is too high, which is why we don't have any drugs, by the way. So it may not be that either one is no good. They may both be good. We may just be asking too much of one or the other of them, and maybe too much of the drugs.
100%.
I think I can speak reasonably confidently to say that if you ask Manal, that's likely where she'd go.
It sounds perfect to me, and I think I love controversy, and I particularly like the last one because I think it's what we see in the placebo if you look at it with the trials. It's not necessarily that the drug didn't achieve its goal, that the placebo overperformed, but they're all in a process, and you have to also judge the process. And if the process affects patients' health, then you look for a different marker within that, because that process improved those non-invasive technologies, it improved that placebo line, it showed an effect. It may not have hit the marker that they wanted because the placebo overperformed, but I can move somebody's liver fat with serial FibroScans every month for 4 months from 290 to 240. Now that is your placebo group way outperforming a medication, but it's the process, it's not. So I agree with Suneil here. I think we have to be, are we asking too much? Should we just be looking for that movement that several of the other presenters discussed in measurements? So I like your controversy.
I'd love to see from the clinical community, particularly the guideline communities, like to put something out there to kind of put some sort of line in the sand so we can at least have a conversation about it. Because, once again, is it asking too much of the NITs? Like if you want 100% sensitivity and specificity, you will not get it.
Suneil, I think the thing that will be asked of the NITs is the indeterminate zone has to come down from 30%. So, one of the things about this being a more practical, less academic meeting was there was more focus on the idea that if you have a double marker, that's not practical. So if we have an indeterminate zone of 30% and then we've only got 0.8s on both sides of it, that doesn't feel terribly efficient. It's better than nothing, don't get me wrong, but it feels like that's a zone that needs improvement. And I know one of the things you and I have talked about offline without going into how or why is the work that you're doing right now and what can we do to slice the indeterminate zone, because I think that's even a bigger question than are you going to get it all right on the sides? You know what I mean?
Yeah, and there's two ways to slice, and part of it's a mathematical abstraction. I don't want to get into like all the like philosophy or whatever, but I could either forget about any particular test. I can take a test and I can say, I'm going to optimize to rule in, I'm going to optimize to rule out. I want to bring those performance numbers as best as I can, and the cost will be I've now introduced this concept of a gray zone. Or I can eradicate gray zones and I can just say I'm going to use a single cutoff. That's all I'm going to do, and I'm going to force a clinical decision. And all I'm doing is I'm going to make all the numbers numerically worse because I'm taking who I would normally say I don't know, and I'm forcing a decision on them. So that's probably a different episode altogether.
Yeah, and it probably is, but it's got to get resolved because indeterminate zone of 30% leaves everybody uncomfortable, and it's simply a question of having minimal values for sensitivity and specificity that you can't hit at the same time.
Right.
And by the way, the rest of the world is a coin toss. If you're doing better than random, you're doing something. Now the question is, how much better than random do you have to do?
Finally, 2 brief discussions from our Day 2 coverage.
In each trial, we have all these non-invasive biomarkers, whether they're serologic or imaging. And, I think we will have very robust data in the near future. And, much of it will be in comparison to biopsy changes over time that will hopefully, if these pharma companies collaborate together, we can maybe move away completely from liver biopsy in terms of trial design and endpoints. My surprise is, why aren't we doing this now? I think, this is a message we need to drive, that we need several pharma companies to collaborate on this because the end result will help everyone in the field. And, we see it in almost every presentation, whether it's MRI-based or serology-based. I think the time is now before we have actually FDA-approved drugs in a couple of years to figure out that pathway in terms of identifying high-risk, juicy NASH patients, as Stephen always says, and then to, figure out a way to monitor them in terms of response to treatment, determine futility, the need to switch to new agents, and how we can prognosticate them. So this is probably my take-home message listening to all these presentations.
That's a great place to start, Naim. And because we're on audio and therefore our audience can't see us, I will tell them that heads were nodding as you were saying that, bobbing up and down, somewhat enthusiastically, in one case quite enthusiastically.
Mine was from a presentation by Shira Zalbayasagi, who did the nutritional epidemiology to practical nutritional treatment in NAFLD. And the thing that surprised me most, and it probably follows on from Suneil last night being controversial, was she reviewed the placebo response in randomized controlled trials and pharmacotherapies and noted that 25% of patients given a placebo improved their NAS by more than 2 points, and that 21% of patients given a placebo improved fibrosis for more than 1 point. So just being slightly controversial at the start, following on from Suneil saying maybe we're expecting too much from the devices and non-invasive, that we're doing, and maybe they're measuring the wrong thing, maybe we should start any placebo response at 21 and above for a trial and accept that as a minimal standard that they've got to tick, which means we prove efficacy on top of that. So we set a benchmark.
You know, Louise, just to say something about this, if you take the baseline biopsies in any trial and then have the same pathologist just read them without any intervention, they'll give you a 20% improvement in the NAFLD activity score. The same pathologist reading the same slides. This is the reality with biopsy. It's a very unfortunate situation we're in, that we have this field, billions of dollars, all these pharma companies, and we're relying on pathologists reading slides. I mean, they're great colleagues. We love our pathologists, but it's not fair to anyone what we are dealing with.
So that's an interesting question, right? So that raises the degree to which what Dr. Zelbersagi was talking about is an artifact of how slides get read versus a reality of placebo. And I'm not even sure how we answer that until we get past slides, but it's an interesting point.
Well, I'll go next, because what I'm going to say sort of connects both of those thoughts. In the first talk this morning, Sven Frank talked about the concept of advanced chronic liver disease, which is fibrosis stages 3 and 4 combined, as being prognostically significant liver disease. And the Bovino consensus, which looks at advanced liver disease and portal hypertension, would allow you to define that using transient elastography. So that begins to push you towards a trial entry point that's defined by elastography and is defined by prognosis, so that you can begin to think about how your treatments will affect an impact on patients in the hopefully not too distant future. So I thought that was quite an interesting idea, and it's the first time I've heard it articulated in terms of trial entry.
I did Anne Daly's genomic and transcriptomic analysis of NAFLD, which obviously follows on from Naim's presentation, but I won't do it as much justice. So basically, she summarised genetic susceptibility to NAFLD, transcription analysis of NAFLD and tissue from biological change to see if we could look at disease progression and can we predict susceptibility to NAFLD. So they did studies of genome-wide association studies, or GWAS. They included over 1,000 cases and suitably matched people. And they took from the FLIT and EPOS studies in Europe. So they had quite a number of patients. So they obviously looked at the modified genes, PNPLA3, they did discover was quite high. They also went for all of the other studies that had shown that GCKR, TM65F2, chromosome 19, and then BOAT7 were identified in their studies as well, but also alcoholic liver disease obviously has MBOAT7. So they went on to look at 1,483 NAFLD cases. They had a central data population of 17,781, which were taken from the UK, Germany and Italy, so they had a good population. And they had 7.4 million SNPs and genotypes. So they found 4 significant signatures with p-values of 10 to the minus 8. PNPLA3, the TM65F2, the HSD17B13, and GCKR. But they had several near misses, but as they got further down, they found that some of these were protective, and the near miss of relevance was the MARC1 and MTARC1 SNPs. So putting that into practice, the subgroup NASH were very much PNPLA3 and leptin receptors for signals, but only the F3, F4 had the PNPLA3 and TM65FS and GCKR. They didn't have the others. So when they went on to the transcriptomics, the biopsy samples had 206 samples. 40% of these were female, average age of 54 and a BMI of 31. But over 55%, just over, had type 2 diabetes. And all fibroses were represented from 0 all the way through to cirrhosis. But the key findings, they were able to find a 25 gene pool that was relevant to these specific patients. And most of those, 24 of those were highly positive and progressive, but only one decreased, and that was the HSD17B14. And this was replicated when they did replication studies. They then went on to the microRNA, which was relevant from yesterday's sessions. And the main microRNA of choice was the 193A5A, which was replicated in NASH with fibrosis 1 and increased in fibrosis 2. So the general comments at the end with all the risk factors are fairly similar. Gene expression changes, specifically at least through progression, there were quite a number of gene changes. And the microRNA was interesting for understanding, but it also could assist in the diagnosis and staging of disease as we progress. So I thought that was quite an interesting topic about being able to, again, genetically identify people who are likely to progress, which may be more relevant when we start to locate these people.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode or the entire NASH Congress series, please send an email to questions@surfingnash.com. Wednesday night, we will post a wrap-up episode from the Fourth Global NASH Congress. We will have a new episode with fresh content next week. I hope you'll join us then. Until then, stay safe and see you on the podcast. Bye-bye now.